Where is it? It is up north near Ocean Reef, probably about mile marker. Just go past.
Yes, just past.
Yeah, just past like that Circle K. Which is a wonderful landmark for a remote view. Yeah, it's on 905. Like if you go past US 1, but after it goes to the stretch, like if you keep going forward, it'll
I
be
don't know where it is.
But if you plug it in Google Maps, it'll pop right up, and that'll take you right there. Oh, Laura's got it.
This planting is part of one of our restoration projects where you may know that Dagny Johnson was originally slated to be a whole big condo development, and there were all these canals that were put in there. We've closed up a lot of them and gotten rid of them, and what we're doing is we're restoring the original hammock plant. So that's what this planting is going to be about. We've got some 50 plants, everything from ground cover to trees that need to go in the ground.
Wear your mosquito gear.
Wear your mosquito gear, yes.
And also we've got our Reef Lionfish Derby and Festival, which will be September. It'll be fishing on September. I didn't change the dates, but it's 9th, 10th, and 11th. Fishing on Friday the 9th and Saturday the 10th. And scoring and check-in and the festival will be on Sunday, September 11th at Postcard Inn. So you'll start to see lots of Facebook posts, lots of social media posts, lots of things about that derby. If you want to register a team, early bird special still going on right now. For $75, you can register 4 people. You all get a sweet rash guard, and you can go out and try to get some lionfish and help remove some of those invasive species. And last but not least, our photo contest is now open. So if anybody has some awesome fish pictures, I know Daryl submitted a couple. Andrew submitted a couple. Definitely throw those in. Go to reef.org/photo contest. The deadline to enter those is July 31st, and they'll be featured on our calendar next year.
And that can be from anywhere?
Can be from anywhere. There's categories, everything from macro to fish portrait, reef surveyor, and action. We have a temperate category this year for temperate fish. So all sorts of cool stuff. So definitely check those categories out. If you've got a great picture, throw it in. If you have a medium picture, throw it in. You never know. And without further ado, let me introduce Dr. Kylie Smith to talk about ICARE.
All right, awesome. All right, make sure that this is working. Found my laser pointer. Awesome. Can you guys hear me on the microphone situation back there?
The microphone is largely for the people live streaming.
Right, just wanted to make sure that he could hear me back there. All right, cool. Good evening, everybody. Thank you guys so much for coming. This is awesome to see this many people in the room, and I especially love some of the ICARE shirts that I've been seeing. So thanks, guys, for supporting us. And thank you guys to Reef and all of the staff that helped put this together this evening. I'm really excited to be here. So as David mentioned, my name is Kylie Smith, and I'm one of the co-founders of ICARE. I'll talk tonight about ICARE, but first I'm going to start talking about some of the work that I did for my graduate research. So I did my graduate research at Clemson University, but I did it down here. So I spent all my summers down here working out on our coral reef system. But then I'll move into ICARE and give you guys updated on what ICARE does and some of the side projects we've got going on. So before I get started talking about corals and why they're so important, and some of the research that I did, I want to make sure that everybody's kind of on the same page about what corals are and why we care about them. So coral reefs are incredibly important ecosystems. They're incredibly diverse. They're what we call biodiversity hotspots. So what does that mean? They support a lot of different organisms. In fact, they support 25% of all marine organisms at some point throughout their life cycle, which is incredible given that coral reefs take up less than 1% of our ocean floor. So the corals themselves provide structural complexity that serve as nurseries for juveniles or protection from predators. And they also can be direct food resources for many of those marine organisms. But coral reefs not only help out marine populations, they also help support human populations. They support they support economies throughout the world by increasing tourism as well as supporting our fishing industries, and they also protect our coastlines. So having that coral reef out there can reduce wave actions that comes in with hurricanes by 84%. So they're really important for protecting our homes and businesses down here in the Keys. Unfortunately, coral cover has been declining since the 1970s, and this is something that we've been seeing throughout the world. In the Indo-Pacific, they've seen a loss of about 5% each decade, and here in the Caribbean, they've seen about 20% loss each decade. So since the 1970s, when we really started seeing coral decline, the reefs of the Florida Keys has lost 97% of our coral. And so we're really moving away from these coral-dominated complex ecosystems and shifting towards these flattened, degraded reefs that are dominated by something called macroalgae. And I'll talk about macroalgae here in just a few slides. People ask me all the time, what is a coral? So corals are actually animals. And so they are made up of these individual cup-like structures called polyps. And these polyps are covered in soft tissue. And you can see sometimes they'll have their tentacles out, and they use these tentacles to capture their food. But you can also see these polyps with the tentacles pulled in. Tonight I'll use the word colony, or maybe you've heard coral colony a lot. Essentially, that's just a collection of polyps. And I'll talk about on the next slide how you get to from a polyp to a colony. And another important characteristic about corals is called a corallite. So as corals grow, they secrete a skeleton made of calcium carbonate. So they're kind of like a rock too. And it's the shape of this corallite, this circular, most of the time it's a circular structure. It's really important because it can tell you a lot about the species of the coral. So the shape of these circles, which is what this soft tissue covers, can tell you whether or not it's a mustard hill coral or a star coral, specifically looking at, are these polyps circular? Are they symmetrical across the colony? Are they raised? Are they sunken into that skeleton? And so that's really important for how we distinguish our different species. So corals may seem like a really simple creature. They may look like a random rock on the ground, but they actually have a really complex life cycle. So they start out as this individual polyp, and they undergo something called asexual reproduction. So they make tons and tons of copies of themselves, and that's how they create a large colony. When a colony gets to a certain size, they'll undergo sexual reproduction, and this is typically during what we call a mass spawning event. So it typically happens once a year. Here in the Keys, it's around the end of August, a couple of nights after a full moon. And they undergo these different environmental conditions, whether it's time of the lunar cycle, different temperature regimes that they go through, and they will start to release their gametes or their eggs and their sperm in these mass spawning events that's synchronized. And so these gametes will meet in the water column, and the sperm will fertilize the egg, and it will develop into a larvae. While the larvae is in the water column, something really important happens. They pick up these tiny little algal cells called zooxanthellae. And these zooxanthellae have a really cool mutualistic relationship with the corals. So these zooxanthellae live inside the coral polyp. So the polyp is providing protection to that algal cell. But those tiny little algal cells are photosynthesizing, and they produce a sugar as a byproduct. And so these corals are feeding on that sugar. And so they're actually getting 90% of their energy from that byproduct. So these guys are really important for helping corals to grow and survive. Another really cool thing that these guys do is they give coral their color. So naturally, coral tissue is transparent. So when you see all your pretty yellows and browns and greens on the ocean floor, that's actually the type and concentration of those little zooxanthellae inside those coral polyps. So these little algal cells are really important, tiny but mighty. And so these coral larvae will spend about 2 to 7 days in this, up in the water column. The whole time they're up there smelling, listening for a happy spot to settle on the reef, where they will develop into a polyp and start the life cycle all over again. So I know that's a lot of science, so I do want to take just a minute to see if anybody's got any questions about that part. All right, cool. I'm seeing nodding, so we're making sense so far. Awesome. So we know that corals are declining, and they're declining for a lot of different reasons. But one reason that I want to focus in on is this local competition coming from something called macroalgae. So we talked earlier about these really important microalgae that have that symbiotic relationship with coral, but most algae is really not great for coral. Specifically, they compete with each other, and they're competing for settlement space on the bottom, nutrient availability in the water column, and several other factors. Unfortunately, these algal species are able to outcompete corals because they are thriving in changing environments that are really stressful to corals. So for example, corals really don't like warmer water, but these algae really love it. So they proliferate and will start to grow and really ramp up that competition with corals. So just like with our coral species, we have a variety of them. We also have a variety of algal species. And so they belong pretty much to three main categories, at least here in the Keys. So we have our fleshy algae up top. These are our Dictyota kind of wide-branched algae that you may see on the reef. These guys are chemically defended. So they secrete a chemical that will suppress the growth of corals. So they're more, kind of chemical warfare in trying to compete with these corals in space on the bottom. We also commonly have a turf algae. This is a filamentous algae, and its defense strategy is just to be really numerous and try to colonize really early. So it's really rare that you'll have bare substrate on the bottom. It's typically going to be covered in this turf algae. And lastly, we have our calcareous algae, and these guys are structurally defended. So they actually have a little bit of calcium carbonate inside their structure. So they're mostly going to compete with corals through shading out corals that are already established, or again, taking up bottom to where those coral larvae can't settle. So in an environment where you have this really strong competition between corals and algae, herbivores can play a really important role. And so here in the Keys, one of the most abundant herbivores that we have are parrotfish. And so there are about 10 species that inhabit the reefs of the Florida Keys. They're super abundant because they are protected from fishing throughout the Florida Keys National Marine Sanctuary. These guys also have a really cool life cycle where they are hermaphroditic. So they start out as females and they'll transition into a male. So these juveniles will start out and they will transition into what we call an initial phase or an adult parrotfish. And then the most dominant of those initial phases will transition into a terminal or a super male phase. And so it's that dominant male that typically is going to establish a harem as well as a territory and defend both of those from other fish or other critters that are coming into their territories. These guys also poop a lot of sand. So next time you're hanging out on a beach, you can think of parrotfish. These guys will go around and they'll graze on rubble on the bottom or coral skeletons. They'll grind it up and they'll poop out sand. So now that you've kind of gotten an introduction to these main groups that we have out on our coral reefs, the overall goal of my dissertation research was to try and understand how these different groups are related. Between parrotfish, algae, and corals, and how they interact on our reef ecosystems to where we can try to identify the factors that are going to maximize coral survival. And so I did this work just off of Layton, so right down the road in the middle of Florida Keys. And the reason that I focused on these sites is because this is very typical of what our reefs look like. They're very degraded, hardly any live coral cover anymore. Any structural complexity that you're seeing is coming from what are called soft corals. Important to an ecosystem, but they're not quite hard corals. And so if we can figure out how to make coral restoration or coral survivorship maximized here, that's something that we would be able to apply anywhere else in the world that are also seeing coral declines. And so the first question was trying to understand the role that these parrotfish play in our ecosystems. So there's been a lot of literature that suggests that these guys are herbivores, that they will graze down the different algal types that are out on our reef, and they're indirectly benefiting corals by reducing that competition. But there's also been a lot of observations of parrotfish directly biting on coral. And these are pretty indicative scars. It looks like someone took a little staple remover and just scraped down the coral. And so the first question I wanted to address is, on our reefs, what are these guys eating? So how do you figure that out? You follow a lot of parrotfish. So for over 3 years, so from 2013 to 2015, my team and I, we followed just about 2,500 fish. We followed them for 3 to 5 minutes, and we noted a lot of what they were doing. What are they eating? Who are they chasing? How many times they pooped? We weren't really sure what we were going to use the data for at first, but then we saw some really cool stories, which I'll talk about in just a second. So we calculated this out. Just a fun fact that after all the time that we spent following these fish between all the divers on my team, we spent just over 5 solid days underwater just making tick marks on how many bites these guys were taking. If you've never followed a parrotfish, here is your chance. So this is a terminal phase red band. And so during this video, you can see he's interacting with some other fish. That's a red band adult. But this is really typical of what you see. They kind of are going over here, taking a bite of something, and then they'll swim over here and take a bite of something else, and then they'll kind of go over here. It's not really very focused biting. These guys didn't really seem to mind us following them. They're very much like Dory, where they're kind of constantly looking over their shoulder like, are you still there? Okay, cool. You're not too close. But I love watching them.
What are the yellow sticks?
The yellow sticks, that was something that we use on our cameras to help us make sure that we're staying a certain distance above. We use this to monitor structural complexity of the bottom. So it's to maintain a certain distance above the reef. I just use the same camera to follow a fish on this guy. So, okay, so we followed all these fish. We noted what they were eating. What did we find? I know, big scary graph, but I'm going to walk you guys through it. So what we have down here, these are called electivity indices. These are essentially preferences. So the more of a color that you see, the more of that particular category they had in their diets. And we have our different categories over here. So we have our 3 different algal types in our greens. And in the oranges, we have our hard coral, soft coral, and sponges. And this gray is other, and that's like out in the sand. So sometimes they'll go out in the sand and pick through for little invertebrates that are hanging out in the sand. And so we have these electivity indices, or these dietary preferences, broken down for each of our 10 species. And so what we can tell so far is we really don't see a lot of those oranges. So at least on our reefs, these guys are ignoring our corals and sponges and soft corals, at least based on how abundant they are in the environment. They're not targeting those guys. But we see a lot of green. So it's pretty safe to assume that these guys are herbivores, and therefore they have the ability to benefit corals by helping to regulate that abundance of macroalgae that we see on our reefs. So that's one question answered. So next we wanted to kind of look at what's going on more of the coral side. And so in response to this decline in coral cover, a lot of different researchers and restoration practitioners have focused on transplanting corals out onto our reefs to help rebuild these ecosystems. But unfortunately, these transplants can face many challenges just like their natural counterparts. These different challenges and stressors are things like competition that's coming from other organisms on the benthic substrate, like our macroalgal species. They also can have direct predation, which we call coral livery, and this can come from fireworms or yellow-footed snails. We also can have increased sedimentation. So when you get particulates in the water, when those settle, it can smother out corals, which is hard for those little algal cells inside to photosynthesize. And they are also susceptible to various disturbances like warming events that can trigger bleaching, of course, physical damage that comes from hurricanes, and just like people, corals get sick. And so diseases can also impact these guys. And so the literature has suggested that there are that coral species differ in several ways. First off, their natural abundance. So we have some coral species like our starlit corals and our mustard hill corals that are kind of more weedy. They're a lot more common now. They've been potentially a little bit less susceptible to some of the disturbances we've had versus some of ours that are more threatened or endangered, like the mountainous star and the staghorn coral. They also can differ in their contribution to the reef community. So the structure that's provided by a bouldering coral can be really different to a little damselfish than the structure that comes from a branching coral. And there's also been some evidence that these coral species can differ to some of those different disturbances that I mentioned on the last slide. And so the second half of my dissertation was really trying to focus on taking advantage of those differences that are naturally occurring between our species to see if we can develop ecological experiments to identify the factors that are increased coral success in our different species using a series of coral transplant experiments. And so I started my first transplant in the summer of 2013, where we transplanted 42 starlet corals and 42 mustard hill corals on 7 different reef sites. So we took pictures of them quarterly through June 2017, and we used these photographs to measure growth of the colony, contact with any competitive algae, so you can see some Dictyota fleshy algae right here. And also we use them to monitor any discoloration that could be a sign of disease or a warming event. And so I want to draw a little bit of attention to 2014 and 2015. So something kind of different happened during these years, and this was a global event, but here's a graph of sea surface temperatures here specifically in the Florida Keys. And so in the late summer, early fall months, we had some severe warming events. And so this triggered bleaching across our sites. So I want to take a step back and make sure everybody knows what coral bleaching actually is. And so if you remember back, we talked about that mutualistic relationship that corals have with those tiny zooxanthellae, right? Those zooxanthellae give those corals their color. So coral, naturally, their tissue is transparent. So remember that. So corals really like their optimal temperatures. And if these corals experience temperatures that are too hot or too cold for too long, they look inside themselves, they see those tiny little algal cells and think, you're not me, you're foreign, maybe you're making me sick. And so they'll expel those little algal cells. And so what you're seeing when a coral bleaches is you're actually seeing the skeleton underneath the live tissue, since that tissue is transparent. If you were to zoom in on these guys, you can see their little tentacles. They're out there trying to feed, but corals are really not good at catching their own food. And remember, those algal cells give these guys 90% of their energy. So most of the time, what ends up happening is these corals die and they become overgrown by algae, and eventually all of this structure will start to degrade. But not all hope is lost. It is possible that if conditions return back to that happy temperature for corals, they can reuptake those tiny algal cells and be able to survive. And so going back to our transplants, so we had these warming events in 2014 and 2015, and we can definitely see impacts of bleaching between our 2 coral species. So we have our mustard hill on the top and our starlet corals on the bottom. And so by watching these, we can see that some bleached completely, but again, not all hope is lost. We can see some healing throughout this time period and then some potential resistance maybe going on during this second bleaching. And we definitely saw some paling, but not quite as much in our starlet corals. And so when we kind of took a step back, we saw that these bleaching effects were definitely species specific. So we saw a lot more bleaching in red in our mustard hill corals than compared to our starlet corals. And we also saw site-specific responses. So it was really more common for a lot more corals to bleach on a site than not bleach. And so we wanted to try to take a look at some of the environmental data that we were monitoring to see if we could help understand these site-specific differences. I know, big graph, we'll talk about it. Essentially what we found, so here are our different coral species. We have our mustard hill up here, and these are the corals that bleached. And what we found is that in these guys that bleached, they had consistently higher abundances of algae, that fleshy algae, directly around the coral. So like literally right around the coral. And so what we think was going on is that corals that are experiencing this higher chronic competition from that algae, they had a higher potential to bleach. So you have just so many energy reserves. If you're spending all of your energy fighting off competition, whenever you are experiencing a warming event, you may not have as much energy to be able to survive and fight off, keeping those algae in you longer. So that's what we think was going on. Like I mentioned before, we continued to monitor these corals through June 2017. Of course, we had some that didn't make it. We had some that survived, but whether or not they survived was not related to whether they bleached or not during one or both of those bleaching events. And so we learned a lot from this first coral transplant experiment. We did see that bleaching effects are site-specific, and we saw that some species are a little bit more susceptible than others. And we find our first bit of evidence that it's this local environment that can really influence the susceptibility to these disturbances. And so like any scientists, we did it again, but we made it bigger and better. And so we added 2 more species. We added, so we still had our starlet coral and our mustard hill coral, but we added a star coral and our staghorn coral. So now we're really starting to get some more of those differences. We're able to compare our more weedy species to our threatened and endangered corals. We're really able to look at bouldering coral versus a branching coral. And as we saw in our last experiment, we already had some evidence that there may be some differences to some of those disturbances. And so the plan was to transplant these guys, monitor them quarterly for 2 years, like in our first experiment, and monitor the colonies for growth, any contact with that competitive algae, and any signs of discoloration that, again, could potentially tell us that there's some bleaching going on. So we transplanted these guys in June 2017. We had 192 total fragments go out. If you guys have outplanted with ICARE before, these corals were a lot larger. The colonies were about yay big. So 192 may not sound like a lot to some of you guys, but these guys were a lot bigger. Our bouldering corals we put down using concrete. Yes, you can use concrete underwater. You just can't play with it all that much. Otherwise, it starts to dissolve. And for our staghorn corals, we put them down using a masonry nail, which we secured the fragment to with a cable tie. So this was in June 2017. In September 2017, Hurricane Irma made landfall in the Florida Keys as a Category 4 hurricane. The eye of the storm crossed over Cudjoe Key, which was just about 50, 60 miles south of where our corals were in this little black box over here. So we were able to come down about a month after the storm in October of 2017 to check out all of our corals. So we have our pre-hurricane here and post-hurricane over here. And our coral transplants you can actually see are in these orange circles. So Irma definitely made her impact on the reef bottom. If you look at the area around these corals, you can see the softer things like our sponges, our soft corals, and that algae. They were completely scrubbed away. But what about our corals? So here we have our transplant survival between our nearshore and offshore environments for our 4 different species. So our starlet corals in red, mustard hill is in blue, mountainous star is in green, and our staghorn corals are in purple. They were outplanted in June 2017. Hurricane Irma is that red bar there, and we were able to come back in October. I was actually pretty pleasantly surprised. We maintained pretty high survival bouldering corals. So offshore was a little bit lower, but you're still looking at, above 60% survival. That's not too shabby. But what we saw in both of these environments was a stark decline in our branching corals and our staghorn coral. And so what we think happened here was that these guys were out there for maybe 2 months. Maybe they really didn't have enough time to really grow and stabilize themselves into the substrate before the storm. But what I like to tell myself happened is that these guys took advantage of their natural life history. So the way that these thickets will grow is they'll grow, have really big branches that get top heavy. So when a storm comes, it breaks them up, they roll away, get wedged in the reef, and they start new colonies. And here is the reason I think this is what happened. After I defended, I was out helping one of my lab mates, and I was able to swim in an area that I had never been in before. And all of a sudden, I was seeing all of these different staghorn colonies pop up. So that's what I like to think happened, is that they just rolled away and they're living someplace else. We're going to go with it. Why not? So we continue to follow these guys over the next year or so. And overall, most of the corals held on to their survivorship. But we do see this green line in the offshore environment. These are our mountainous star corals. We see this big decline all the way through the next quarterly sampling periods. And so we started to look at our environmental data again. And what did we find? We found that this fleshy algae, these corals that died, had significantly higher amounts of that competitive algae. And so again, we think that this is some evidence that when they're dealing with high competition in their environment, may just make them less able to survive whatever stressful environment that they're dealing in. After a hurricane, with all of that stuff being scrubbed away, there's a lot of stuff that's changing in the environment, a lot of things that are trying to come back. And so that's what we really think was going on, is that they're using up all the energy to try to handle everything else that they just get overgrown and outcompeted. And so we were able to learn a lot throughout my graduate work. Looking at our different coral transplant experiments, we did see that not all coral species are the same. They can respond differently to different stressors and different disturbances, but we find a lot of evidence that it's these local conditions, and I mean like literally the half meter around a coral colony, that can impact whether or not these guys can survive. And so our goal was to try to use what I learned based on what parrotfish are eating and how they're acting, and also looking at the community around our coral transplants to try to use that going forward to inform how we're doing restoration, to try to put these guys in areas where these corals are going to be the most successful. So before I start talking about ICARE, there's a couple more steps I need to get through. I have to thank all of these people that helped me do my graduate research. Clemson University has an awesome program called Creative Inquiry, and it partners undergraduates with graduate and faculty researchers, and it really immerses these students in science. It teaches them how to do it from the ground up. We were able to bring a lot of these kids down to down here, get them certified, let them fall in love with the ocean and these systems, and they were able to do a lot of this data collection. But more importantly than that, it taught me that anybody can help make a difference. All you have to do is give them the education and give them the opportunity, and they will completely amaze you with what they're able to do. And so that's a really big part of what we do at ICARE. So as I talk about ICARE, I have to talk about this man right here. So I'm sure many of you recognize him. This is Michael Goldberg. He is my co-founder of ICARE, and he's back there in the room blushing. So woohoo, I got him to turn red. So Mike is the owner of Key Dives. And so Key Dives was my support shop while I was doing my graduate work. They filled our tanks, they serviced all of our gear. And every time I went into that shop, Mike would come running out of his office and he'd say, "Hey, I saw this on the reef today. What is this? What's going on?" And he is the most passionate individual that I've ever met, but he's been diving down here for many years. And so he has some observations that I don't have. I was down here one December, it was December 2018, and I was down here for a conference, and I met up with Mike and some of the Key Dive staff at the Florida Keys Brewing Company, and we started having this conversation about what I was learning through my graduate work, and he started talking about some of the conservation work that they've been doing over at Key Dives, which if you don't know, Key Dives has an amazing record for doing conservation, specifically through their marine debris dives. They have removed over 15,000 pounds of marine debris from the reef using only diver hands, which is amazing. And so he's talking about these passionate people, these divers that are coming and wanting to be a part of the solution. And we just had this idea over a beer in the beer garden. Why can't we combine these two ideas? Why can't we bring the science to this army of recreational divers that want to make a difference and put coral restoration in their hands and let them be an active part of the solution? And so that's really what we do here at ICARE. So ICARE stands for Islamorada Conservation and Restoration Education, and we are the only reef organization focused on restoration just down the street in Islamorada. So why Islamorada? Why do we care? Islamorada has some amazing fish life. Just take Alligator Reef, for example. You do a dive there, you're pretty much guaranteed to see turtles, sharks, eels, hundreds of species of fish. But unfortunately, the coral's really not doing so hot. This is a mountainous star coral at Alligator Reef in 2015. This is that same coral head in 2019. Yes, it is sad. But what's even more sad is that people are jumping in the water and they're like, "Wow, look at that coral head. It's gorgeous." So we've really realized that there's a gap. There's a gap in the education of what people's baseline are, not knowing what a healthy reef should look like. And so we're all about educating the public, as you know, tourists, locals, educating people on what a healthy system should look like. There are a lot of organizations that do some amazing coral restoration work here in the Florida Keys. And one that I have to talk about in particular are our partners at Mote Marine Laboratory. So Mote, they are fantastic. They provide us with all of the corals that we transplant, and they do some amazing science behind the corals that we're outplanting. So where do they get their corals? They go out to the reef and they take samples of coral colonies that are still alive, which means, let's just take the last decade or so. They survived back-to-back bleaching events of 2014 and 2015. They survived Hurricane Irma. They survived stony coral tissue loss disease and everything else that has been thrown at them. They take a sample of those corals, they bring them back to the lab, and they cut them up into tiny little pieces, about the size of your smallest fingernail. They then take them and they glue them onto these ceramic pucks and they put them in a nursery to grow. So these guys are going to grow much faster because they've got kind of a raw edge. So think about it of your skin. If you cut your skin, it triggers a growth response and it tells your skin, I need to heal faster. It's the same thing in these guys. So they can grow to cover these ceramic pucks in anywhere from about 2 to 8 months, depending on the coral species you're working with. Some species are going to grow faster than others. They then take these fragments and they put them in experimental conditions. So they'll ramp up the temperature, they'll drop the pH to levels that they anticipate will be on the reef 30, 40 years from now. And it's only the corals that survive these experimental conditions that are transplanted out on the reef. But they don't stop there. They do something that's called assisted reproduction. So they will take a strain of coral, or a genotype is what we call it. Say this strain is really great at dealing with high temperature, and this strain is really great at dealing with, say, low pH. And they will cross those different genotypes and create new genetic diversity that have the traits from both of those parent colonies. And so they've been able to create, I believe, just over 1,500 different genotypes that hopefully are going to be more resistant to what the world is about to throw at them. So it's not just transplanting corals, it's really trying to transplant corals that have the highest chance of survival in our future.
How do they cross? So do they put them together or just from spawning?
I actually don't know if I know that part. They do have, I know they have a system now where they can replicate spawning conditions, but I don't know if that's how they started it or if they started it doing, like waiting for the natural spawning cycle. I don't, there are some Mote people here in the audience, but I don't want to, you know, we'll talk after. So even the way that we outplant these corals, so everything we do for outplanting corals, we do follow Mote's protocols, but even the way that they put these corals out is incredible. So for bouldering corals, they do something called coral skinning. So they take advantage of these old dead coral skeletons. We go out, we scrub all the algae and sponges off of them. We'll move the sponges someplace else. They'll grow someplace else. And we'll glue these little pucks onto these coral heads. And as they grow, they're going to grow faster because they don't have to secrete a skeleton. They're literally able to grow their tissue over top of the skeleton and skin the coral. And all of these pucks are coming from the same parent colony. So as the tissue grows and it meets, they'll recognize that that's itself and they'll fuse. And so they're able to get a much larger coral colony much faster than they would a natural colony. So this is a coral in 2 years. Naturally, this can take anywhere from 15 to 20 years. Not only are these guys out there helping to contribute to the reef that we have out there, they're providing structure, they're helping to support those different marine organisms, but these guys are also contributing to the future populations. So about 3 years ago during coral spawning, the Mote scientists saw the first coral that they put out using this skinning process. This guy was 5 years old. That is awesome. I love it. Yes. In this video, you can actually hear the scientists like screaming underwater because this is amazing, right? So in just 5 years, they're able to get a coral to spawn naturally. This can take anywhere from 50 to 75 years, depending on the species. So this is a huge steps to be able to get these corals to start sustaining themselves. So that's everything that we do with the bouldering corals. We also work a lot with our Staghorn branching corals. And the theory behind where they come from is very similar. They go out to natural colonies that are surviving. They take clippings, put them in these coral trees in the offshore nurseries where they grow. And when they get big enough, they go out and they take clippings, which they give to us. And then we transplant them in these clusters of 5 using a masonry nail and a cable tie. Again, all of these clusters, or the fragments in this cluster, came from the same parent colony. So as they grow, they'll fuse and be able to create a larger colony much faster. So what do we do here at ICARE? What makes us a little bit different from some of the other coral restoration organizations? So we partner with local dive shops and we train and educate their customers to be a part of coral restoration. So they come to our training session, we teach them everything we know. I say we, it's my team of amazing interns that teach divers what they need to know. And then in the afternoon, we put coral in their hands and we let them be an active part of the restoration. So about 95% of the corals that we put down at ICARE are done by recreational divers. And this has been a great way to educate visitors and tourists alike about the status of our coral reefs and what people can do to help. We also, again, as a part of our partnership with Mote, they've established the first and only land-based coral nursery. So we base out of Bud and Mary's Marina, just down the road in Islamorada. And so we include tours of this nursery in our educational sessions that we give to all of our divers. But this is also open to non-divers, so you can see a little bit of that coral husbandry side. Another thing that makes us different is we monitor and maintain 100% of our coral transplants. So when you get a permit from the sanctuary, bare minimum is to monitor 20%. And so we actually followed Mote's example and we monitor 100% of our transplants, but we do a lot of maintenance as well. So we're going out at 1 week, 1 month, and annually for 3 years to check in on all of our coral babies. And so if you are a local, we do have a new program that we got funding to help support locals coming out and helping us do this monitoring and maintenance program. So if you are interested in finding out how you can dive for free and help our little babies, you can talk to me, you can talk to Mike. There are a bunch of ICARE reps in the audience tonight. The last thing that I want to talk about that makes us different is I had to stay true to my roots and monitor the reef community. So we do annual surveys with our team of staff and interns where we're monitoring natural corals, disease prevalence. We're monitoring the invertebrate communities, so our lobsters and crabs. And we're also looking at some of those other organisms that are on the benthos, like sponges, soft corals, and of course that competitive algae, to help us understand where and why our corals are doing better. And so where are we? So we've been outplanting corals here since January 2021, so about 1.5 years. And to date, we have outplanted just over 5,700 corals from 4 different species. We're focusing in on 4 different reefs that range from shallow patch reefs to a deeper spur and groove reef, again, so we can kind of get an understanding of who's going to do well where. And our survival rate at 1 month post outplanting is 88%. And we've had just over 1,100 individuals come through our program. It's 1,100 divers that have been educated on what a reef should look like and get corals in their hands to get them involved. And so some of our successes, if you go to a site called Rocky Top, this is our crown jewel. Our corals are amazing there. So these are some of our staghorn corals. You can see here where the cable tie is. You can hardly see the cable tie. The coral completely grows over it and incorporates that into its structure. It'll grow onto the bottom, and these guys will fuse together and become one giant colony. We have a couple of these thickets there where we look down and you can't even tell which was that original 5. They've all just fused together, and they've been out there for less than 1.5 years, which is so exciting. Another kind of success story that we weren't super expecting. We've also been working with Elkhorn Coral. We've started doing this a lot here recently. These guys are put down on a puck just like our bouldering corals. They're put down with an epoxy. They get hammered during their first week. They get a lot of parrotfish predation. They go through some weird color changes, but then they pop back after like 1 or 2 months and they are gorgeous. They fuse together, and this guy is 1 year old and they've already started to get these little crowns coming up. So we're excited to work with more and more of these guys. So we have a lot of great partnerships and collaborations. I'm a firm believer that you can't do everything well. So I do want to highlight just a few of them. I've talked a lot about Mote tonight, but I wanted to give all this information. If you are interested in seeing the land-based coral nursery, you can come to our educational sessions. We have one every Saturday morning at Bud and Mary's from 8:30 to 10:30, and we will include the nursery in that educational session. If you would like to come and you're not a part of one of our dive trips, we do ask that you call Key Dives and reserve your spot. It's all free. We just have to make sure we put enough chairs out. We also have a great partnership with Reef. So we do monitor members of our marine community, but I am not a fish person except for parrotfish. So we have our reef divers come out and do fish counts on all of our sites. So they can give us estimates of fish diversity and abundance, and then we can relate that back to our outplanting effort and how our sites are changing through time. And what we're hoping is that we'll start to see some increasing in some of the different fish species that we have. Another cool project that we've got going on is we are restoring sponges. This is a collaboration with a graduate student at Florida State University, Bobby Renfro. I used to think that all sponges were awful competitors with corals. They were bad guys. And I have learned so much. I've learned that there's only about 2 or 3 species of sponges that are actually competing with corals. These sponges act as glue. So they're helping to stabilize structure that will allow for more areas for corals to stay, or for corals to settle, and for more area for us to come through and transplant corals. And so what we've been doing, it started as a pilot project that's now been picked up and funded for the next 2 years, where people can come, they learn everything they need to know about sponges, and then in the afternoons, they can come out and transplant sponges onto our restoration sites. So if you didn't get a chance to do that this past summer, Bobby's coming back next summer, so sign up for our newsletter so you can find out that schedule. And the last project that I wanted to talk about was how we're trying to help our herbivores. So I talked earlier about how herbivores can play a really important role in reducing some of that competitive algae that we have on reefs. We have a lot of parrotfish, but sometimes they need some help. We have some urchins, but they're struggling a little bit right now too. So another really important herbivore that we have is the Caribbean king crab. So this is a collaborative project that we have with another scientist at Mote, Dr. Jason Spadero, where we are trying to enhance the populations of these herbivores. So I say we, again, it was my wonderful team of interns. I was on the boat, went out and did a night dive through the channel at Bud and Mary's Marina, which is kind of sketchy. You guys are all awesome. Thank you for doing that. And we also did a dive out by Chica Rocks where we went out and collected adults. So we were getting our males and females. Sometimes the females would have eggs, sometimes not. We take them down to Summerlin Key, the Mote facility down there, and they are breeding like crazy. They have hundreds of crabs down there that we are growing up. And the overall plan is we're working with the FWC right now to come up with a health check protocol. But we're hoping that over the next couple of months, we'll be able to release these guys on our restoration sites, and we'll be able to monitor their impact on the algal abundances that we have there. So you may be sitting here thinking, how can I get involved? There are so many ways you can get involved. If you're a diver and you're interested in coming out and transplanting corals, you can book a dive with our partnering dive shops. We run a trip every week. So typically they're on Saturdays. Sometimes they're also the 3rd Sunday of each month. And so our participating dive shops are Key Dives, Islamorada Dive Center, Conquer Public Divers, and Jake's Offshore Adventure Charters. If you're not a diver, but you really like getting in the water, you can come out with Islander Girl Snorkel Tours. Every Friday, we put a staff member on the boat. They go out and they jump in the water with you, and they'll point out and let you know what everything is. If you are a reef diver, which I'm hoping some of you are, you can always come out to our sites and help us count fish. Following us on social media is a great way to get caught up about what opportunities that we're offering. We are on Instagram and Facebook at I.CARE About Coral. And if you're interested in finding out more information about our side projects, we do have a monthly newsletter that you can sign up for at icareaboutcoral.org. So I really like the word community because for me, it has so many different meanings. We are doing this work by the community. We have an amazing community of divers that come out and do this work with us to be a part of this solution. But we're also funded by the community, and we've been incredibly blessed and amazed by the different places that this support has come from. But we're doing the work for the community, not just the reef community, but also the community of people that we have here in the Keys, to try to make sure that we have a future. Every single person that lives in the Keys is directly or indirectly impacted by that reef. And so if we don't have a future on that reef, we don't have a future here on our islands. So I do have to take just a couple of minutes to thank our contributing partners. These partners have come in all different shapes and sizes, from dive and snorkel operators, fishing charters, restaurants, businesses. We have a tattoo artist, we have realtors, we have lawyers, coming from all over the country that have been supporting what we do. We also have a couple of different private foundations that have helped us out, as well as the city of Islamorada has been giving us some funding. So we are incredibly grateful. They've been incredibly creative in how they are supporting us. Some support us by donating percentages of their overall revenue. You can go into shops around town and see our donation boxes, or you can see some of these really awesome I Care About Coral t-shirts that are in the audience. So some of our merchandise is being sold around as well. So a huge thank you to everyone that helps us support all of what we do. So that's all that I have for you guys. Here is our contact information. If you're sitting there thinking, I want to get involved, I don't know how, or you've got a cool idea, feel free to shoot us an email, and we'll be happy to chat with you. And I'll take any questions. You guys are great. Woo! Yes.
So you said that they clean off all the algae before you start. Do you do that after you plant as well, or do you just kind of let it go?
That's a great question. So the question, so everybody at home can hear, was, so we clean the algae before we transplant. Is that something that we continue to do? And yes, we do consistently clean algae. It's part of the maintenance that we do. So we'll do it. 1 week is not usually needed, but we're constantly going back to those same 4 sites with our little scrub brushes and trying to scrub that out. Yes.
How does brain coral as a species fit in with these other corals, and is anybody doing any planting of that species?
So the question was how brain corals fit into kind of an overall restoration plan. We are getting ready to start. Actually, we're picking up our first batch of brain corals tomorrow. So yes, there are species that people are working with. My understanding of whenever you frag them, because the polyp structure is so different, it's more of like those series of ridges as you go across the coral head, that they can be a little bit harder to fragment. So I think it was a little bit slower going than like our circular polyps. It's easier when you're cutting to see where those polyps are separating. But they are starting to work with those, as well as they're starting to work, like the Florida Aquarium is starting to get some of them to spawn and create new babies that will hopefully be coming back down. Yeah.
Any data on food source for people eating parrotfish? I've been to restaurants where they have them in a, I mean, we love eating lionfish because they're bad guys, but people eating lots of parrotfish.
So the question is about data of people eating parrotfish. I know that there are places throughout the Caribbean that do eat parrotfish. That makes me super sad. I know that people will collect them for the aquarium trade, but I don't know a whole lot about people doing that.
Question, divers, when you see a beautiful chunk of coral living, thriving, rolling across the bottom, is there any hope to stick it in the sand and say, grow, baby, grow?
So the question was, is if you come across a big coral head that's alive out in the sand and it's rolling, the best thing that we can tell you to do is to call us, because we would really love to be able to come out and help right those corals. But there can be a lot of issues with that. Of course, the sanctuary. So after Hurricane Irma, the sanctuary came out and said, anybody that has a coral permit, if you see a coral that needs to be righted, do it. But we just want to make sure that it's done in a way that maybe it'll be stabilized. Because one thing, actually on one of our sites, the last tropical storm that came through, it cut off one of our coral heads that had some of our coral transplants on top, and it flipped it. What we wanted to try to do is stabilize that to where during the next storm, it's not going to cause more damage by rolling into our other transplants. So there can actually be a lot of work involved, especially if it's a big coral head, to try to get lift bags in and move them.
Chance of survival probably minimal.
Chance of survival for those, it depends on how long it's been upside down and in the sand. I think they can probably survive, I would say, again, depending on the species, maybe 2 or 3 weeks at least, maybe. Corals are pretty amazing. I mean, they're pretty fragile, but they're also pretty amazing and can come back. Yeah. There you go. There you go. Any other questions? Yeah.
Do you have any comments on water quality and root causes?
So the question was comments on water quality and root causes. Not being a long-term Florida native, I'm not as intimately involved and aware of some of those. What I can tell you is that whenever I first started working down here, I started working in the Florida Bay a lot, and it was consistently green. And then whenever I moved down here in 2019, I started working for FWC. We were doing sponge restoration in the Bay. I had seen major improvements. I think we still have a lot of ways to go, but I think that there are some things that are trying to be done to improve water quality. And I've seen noticeable improvements, but I can't really speak a whole lot to those causes that are down here. Anything else? Yeah, sorry.
Okay, going back to parrotfish. So in the study of land animals, often you dictate what they eat based off of their teeth, whether they're carnivores, omnivores, or herbivores. So did you have thoughts on the very obvious human-like herbivorous teeth of the parrotfish before you started to study what they ate?
So the question was, can you look essentially like at their mouth structure to determine what they eat, kind of like in land animals, if like molars or like grinding teeth versus carnivorous teeth? Is that?
Yeah, like canines, like if you looked at a barracuda versus a parrotfish, very different teeth.
So not really. It kind of. Okay, we're going to go with kind of. So there are species. It's not necessarily the shape of it. It's more the robustness of it. So there are species like your the scarids, which are your rainbows, midnights, and blues that have that like bigger jaw morphology. And it's actually the muscles that are behind there and like can really help with that scraping. So there is some evidence that these different parrotfish, based on their jaw morphology, can fall into different categories in terms of how much of the substrate they're removing when they take bites. But it doesn't necessarily tell you, so like blues, for example, they're one of the biggest ones that are out there picking through the sand looking for little invertebrores. But if you were to look at that beak, you would think maybe they're really excavating bigger pieces of the substrate. So yes and no, but it's not always clear cut. They've done a lot of isotopic analyses where they've taken tissue samples and tried to look for profiles of different algae, different corals. And because these guys are such generalists, they're biting here, biting there, biting there, they just can't do it. And the same thing with like gut content analysis. There's so much in there that they eat everything. Yes, Mike. Hi, Mike.
We see a reduction in the parrotfish population, especially in some of the ones that do most of the algae.
It's been a little bit. It's been a few years since I started taking, or since I was taking those parrotfish numbers. So we have at Clemson, they have continued to count parrotfish on the sites, the 36 sites that I established. During my tenure there, we did see a decline in some species, but it bounced right back up. So we think that, it could be one of the sites that we saw it at was a site called Coral Gardens that is kind of similar to Chica, where you can't spear. And so we started to see a lot more lionfish there. We definitely see some seasonal trends. We typically saw less parrotfish during the winter, whether or not they're moving deeper where it's a little bit warmer, or they're just not moving all that much because they're cold. But we really weren't seeing a huge trend one way or another. You would see differences in species depending on where you go. Alligator Reef is the only site that I monitored where you could see princess parrotfish out of all the 32 sites that we looked at. 36. So.
I've got all sorts of questions.
Oh, great.
Thank you so much, Kylie.
Thank you guys so much.
All right. Thank you all so much for coming. Kylie, we really appreciate all of your hard work for Reef and for ICARE. Yeah, so thank you all so much. We really appreciate it. One thing I forgot to mention earlier is definitely register for Reef Fest. You can dive with Key Dives then. You can dive with several of our local dive partners. It'll be October 13th through 16th this year. So make sure you register online. It's a free event, so check it out. And thank you all so much for coming. Our next Fish and Friends will be the second Tuesday of August. I believe it's August 9th. And it'll be Sarah Fangman, who's the head of the Florida Keys National Marine Sanctuary. And she'll be talking a little bit about the restoration blueprint. So that is one you will not want to miss. I'm sure everybody has lots of questions for her. So put that on your calendar. And we will see you all soon. Thank you so much for coming. We really appreciate it.